official scientific director from the International Rett Syndrome Foundation. And, to me, most important, she is a Rett mom, if you did not know that. Daughter is 16 now. Catalina is 16. She has roots here. She interned at Gillette. She interned at Gillette, so she has a history with Dr. Bising. We met her, I know we met her way back at the walk and met Catalina, and she was probably 3. So, and here you are. You are lots of expectations. So, you're really smart and you're a mom. So, what a great combination. So, pressure on.
Well, thank you. Thank you to Lisa and to everyone for inviting me to be here. It is really an honor to be back here. As Lisa said, I have my roots here. Catalina was diagnosed, and we went to the Gillette Clinic in 2008, so I guess soon after it had started. So thank you for starting that clinic so that I would have a place to bring my daughter when she was diagnosed and I was doing my residency out here. So it's really an honor to see all of you that I know from previously and to meet many of you that I didn't know. So really, I'm here to talk about where is research. We spent the morning talking about trofinetide, and I'm really not going to focus on that because where do we go from here? What is left to do? And to start off, I'm going to just show you my why. So this is Catalina. As Lisa said, she is 16 years old. She's in 10th grade. She is a joy. She's very social. She loves being with her friends. She is in general education this was homecoming, that bottom picture. She really thrives in her current environment. And I bring this up to say, every one of our kids and every one of our family units is different, and what we do and how we do it has to be driven by them and by us. What fits into our lives? What can we manage, and what can our child manage, and what is it that they want? She always wanted to be in the general education setting. So we have fought for her to be in the general education setting. She's going to graduate from high school with a diploma and go to community college, if we can make it all work. Because that's what she wants, and because we've been able to fit it into our lives. There were times where we did 40 hours of therapy a week, and there were times where she has had no therapy because it didn't fit into our lives. We're a family unit. There's like the best practice of what I think should be done, and then there's the day-to-day reality. And I think for all of us, we have to take that into account when we're talking about research participation, when we're talking about clinical care, when we're talking about clinical trials, when we're talking about newly approved FDA drugs. What fits into your life as a family unit and into your child's life? What is it that they want? All right, so with that, it's amazing that there are 13 families, you said, that haven't been to this particular Ed Day before. And so I really, for those of you who've been around for a while, you know the history. And for those that are newer, we are where we are today because of decades of research. And so I put this up. This is actually an old figure. It stops in 2016. So this was a figure that showed 51 years of progress from the time that it was first described in the literature in 1966 to where we are today. The gene was identified. The animal models were made so that we could actually study Rett syndrome and understand what this gene and the protein do, and we could test treatments on it. We started clinical trials. We started the natural history study to learn more about it. And then, really in the last 5 years, we've added to this. I'm not good enough to make new graphics, but I can make bullet points, so I add bullet points. But really, in the last 5 years, look at what's been accomplished. In 2019, the first phase 3 trial for trofinetide was started. And I will say, a phase 3 trial for a rare disease, to us, it's kind of like, meh, it was a phase 3 trial. For those that have been around, when Catalina was diagnosed, there were 0 drug companies with any interest in Rett syndrome. We talked about small trials in academic centers. There were no companies that were going to develop drugs, right? So in our lifetime today. So we had a first phase 3 trial. At IRSF, we launched a new Center of Excellence designation. We really looked at our centers of excellence and said, what is a center of excellence? We want the centers of excellence to be experts in the clinical care and experts in research. Because where we are today, we need both. And we're thrilled to have partners like Gillette to be one of our centers of excellence. We have 18 centers of excellence in the US now. We started a new registry. You heard earlier today about the natural history study and how important that was. So that was a longitudinal study over 15 years that the NIH funded to really learn what is Rett syndrome, what can we expect, what happens, what's the course of the disease, the course of the condition. And that was fantastic. Funding ended in 2021. And we said, this condition is continuing to change because we're identifying new people that are on either spectrum, either more severe or less severe than what fits into the clinical criteria. We have more enriched environment these days, so our kids are maybe getting more access to therapies younger. We have drug trials and now an FDA-approved treatment, so this is going to change the course of the disease. So we need to continue to study that natural history so that as we develop new therapeutics, we continue to have a current view of what that baseline is. That baseline will include trofinetide. It is an FDA-approved treatment. We need to know what is baseline for people on trofinetide so that when we test new therapies, it compares to what will become standard of care for those that are able to and choose to take it. So that's why we launched the new registry. And for the registry, you have to register through one of your centers of excellence because it's really important to get that clinical perspective because that's what happens in clinical trials. The rest, so that Acadia submitted the new drug application in 2021 or 2022. The first gene therapy trial for adults with Rett syndrome was started in Canada. Now we've heard of the first gene therapy trial for kids. That's going to happen in the US. And then, of course, we've had the FDA approval. So this is huge, like where we've gotten from a disorder first being diagnosed to where we are today. But how did we get here? 6 decades of research. Right? This didn't come overnight. None of this happens as fast as any of us parents want this to happen. We want our kid to be diagnosed and treated tomorrow, or really treated yesterday before they regressed. But it's taken 6 decades to learn about MECP2, to understand how can we treat it. You heard Dr. Feynman talk about this BDNF and this IGF-1. Well, the reason that they know that those would be potential targets is because of the research that was done, that research that's done in the lab. There's been more than $800 million in funding in MECP2. And this is really critical. The federal agencies are big funders. Rett gets, $800 million is kind of like nothing compared to other diseases, right? This isn't, big major diseases. But without the NIH and the DoD putting money in, we wouldn't be able to make some of the big, basic accomplishments that allow us to get to where we are. And then it's the funding that comes from more patient advocacy organizations that really says, okay, but we need Gillette to have this. Because NIH isn't going to give Gillette money to improve the care of your kids or improve the research here. And so it's really a combination. It takes the federal funds, but it also takes the more Rett-driven funding. And that's why it's really important to have all of this. We got here because we set the stage for pharmaceutical companies to come in. We now had animal models, we had possible targets, we had a gene that they could target, we had a community, we had doctors who were familiar with it, we had a natural history. This was all part of what got us here today. The doctors who are also researchers, they're not only clinical care, they get to wear that hat, and that's a huge part of what they do, but then we also say, and please be a researcher. Please conduct clinical trials. Oh, and in your free time, why don't you create some studies for, understanding what diarrhea is in trofinetide, right? Just in your free time. We have hundreds of families giving their blood, sweat, and tears. And I will say, these families, we've already highlighted some of the families that participated in the clinical trials, but it's also the families that say, today's not the day for a clinical trial for my child, for my family unit. I can't do it, but I can do a survey, or I can come back to clinic, or I can say, not this year, but I will next year. And all of those are fine. And all of those help to contribute. So each one of us help to get us to where we are today and where we're going to be tomorrow in whatever way works for us and our family unit at that time and our child. Dedicated families who run volunteer organizations like the Midwest Rett Syndrome Foundation, really to bring you guys together and to help organize so that you can get your local needs met and those local needs can be heard on a national level. You know, we want to hear from all of our families, and it's organizations like this that really are critical to the success of Rett syndrome care and Rett syndrome research. And then IRSF really helps to set the stage by working with not only the basic science researchers, but also the clinics to make sure that we're headed in the right direction and looking at new treatments. So where do we go from here, right? So that's everything that got us to where we are, and we have so much further to go. I'm so excited about having one treatment, and I will be even more excited when we have multiple treatments and multiple different approaches. And so really, this is just, I'm not going to go through these bullets, but we need to continue to build on what we have. We have so much momentum, and I think that where we are today sets us up really nicely to get us to where we need to be. But we need to continue funding basic science research to find new approaches for better drugs or different drugs. No one drug will work for every individual. Not every individual will qualify for gene therapy. Not every individual will want a particular drug or a particular gene therapy. And so we need multiple treatments. Dr. Fima had said, he said on average, any drug that he prescribes for seizures works on 30, 40% of the participants. We'll probably expect the same for Rett. It's a complex disorder. No medicine works for everyone. So we need multiple treatments. And it really takes all of this system to get us to where we need to be. Some of you may have seen something like this. Some of you may be sick about hearing how long it takes, but it's the reality. None of us wants to hear that it takes a long time, but it takes a long time. And I think the more that we're prepared to understand that and say, we're grateful for where we are today, but look at how long it takes. The basic science research on a particular drug could be 5 to 10 years. So that's what's going on in the background all the time. That's where we're looking at the research, we're looking at the researchers, we're trying to identify new basic science research to get us towards new treatments. Translational research, that's going from the lab, looking at things into, getting closer to a drug. So you might be in the animal models, testing how particular treatments are working in animal models, usually in the mouse. And then the clinical research. And that trofinetide trial is a perfect example. It says 3 to 10 years for clinical research. That's pretty standard because you have to have multiple clinical trials. It's not just one clinical trial. And I have on a slide coming up, the first trofinetide trial started in 2013. So 10 years to get us from the first clinical trial to approval. These next two graphics are, again, just to kind of drive home the point of why we need multiple approaches. So what this first figure is, you see, so HCV and MRSA are two infectious conditions, and then Alzheimer's, as many of you know, it's a neurologic dimension. So this is not Rett specific. We don't have graphics like this for Rett. But really what it's showing is how many drugs or how much money do you have to start with, how many drugs do you have to start with to get down to one approved treatment. And what I want to kind of highlight is for Alzheimer's, your overall success rate is 0.5%, right? So when you start in the lab and you're like, oh, I think that this would be the right approach. And then you say, okay, there's a whole bunch of drugs. This next figure. There's 10,000 compounds that target this particular, I'm going to use IGF-1, for example. Well, now you're going to say which one of these 10,000 compounds would actually cross the blood-brain barrier, be able to be taken orally, blah, blah, blah. You get down to 250. Then you say, okay, which one is actually your lead compound? You might get it down to 1, 2, 3. This graphic shows 5. And then at the end, you get 1 approved treatment. When you're lucky, right? And so it takes thousands and thousands of possible ideas to get down to your 1. Again, this is just to highlight why we need that continued research, because we need to keep finding those 1s. And so it's going to take hundreds to get to the 1. Not that we have to test all hundreds, but we have to do the research, the early research, so that we can get to the 1s. And so how are we doing it? So this figure here is all of the researchers that are currently funded by the International Rett Syndrome Foundation. So as of today, currently working on Rett syndrome. So these are researchers that are working to look at the basic function of MECP2 in the very, very early development. So pre-regression in the animal models through regression to see what exactly is changing and how can we modify that. To researchers that are studying RNA editing, to researchers that are looking at new tools to use in the clinic to measure changes, to researchers that are studying what is the course for males with Rett syndrome and how does that differ for females with Rett syndrome. So these are researchers that are dedicating. These are multi-year grants. And we do grant calls every year to add new researchers to this, and that takes funding. And it's critically important to get us to those new therapeutics. We were really fortunate this last year to be able to earlier this year, make our largest investment in history. So we were able to fund $4.4 million in research that spanned from basic research through to clinical. And this is a trend that we want to continue because the more we can put in, the closer we'll get to having those multiple therapies. And this is just one figure. These are the newly funded researchers. We did a 2-day symposium, and this is part of what we do. We want to bring the researchers together to get there faster. So all of our newly funded researchers, now we do a 2-day symposium. They each present their research studies. This was about 3/4 of them. They weren't all there on the image. And they talk about their research, and they actually learn from each other. Oh, you should actually consider doing it this way. You should do it that way. And so it's going to improve our studies when we have that collaboration across these people that some of these researchers have never done Rett research, but they're an expert in RNA. And we want to better understand that aspect so that we could develop therapeutics in Rett. And so we bring them into the field. And this is what allows them to have the other experts teach them so that they can make their research more valuable. And then we hold an annual scientific meeting. And so this will be in June of this year, again, to bring those researchers together to get us moving faster where we can to decrease that timeline. So you saw that initial timeline. The role of foundations like ours is to decrease that timeline so we can get there faster. I said this earlier, and I really, truly believe it. I want us to be at a point where you have a medicine cabinet worth of treatments. You have multiple different approaches. They have different targets. Some of them are genetic approaches. Some of them are what we call small molecules or the drugs that you take. And so let's talk about some of these clinical trial updates, really what we want to hear for what's current research going on in clinical trials. This is a graphic that we have on our website, and it's gone out in some of our mailers. I want to remind everyone, in 2008 when my daughter was diagnosed, this wouldn't have existed because there were 0. It would have been a very easy graphic. 0 companies invested in Rett syndrome. And now we have companies that are at different stages. And I think that this is really reassuring. No, they're not all in phase 3 trial. No, they're not, all FDA approved. But even just to have this many different companies, all with different approaches, that are investing their millions of dollars to develop their treatments for Rett, not all of them will be successful. We know that's the reality. That's why we do clinical trials. If everything was successful, we wouldn't have to test it in a trial. So some of these will not be able to move forward. But the more of these we have, the better off we'll be. All right, so more specifically, we've talked a lot about trofinetide. As I said, I'm not going to go into it, but only just, I put on here the dates of the the trials, just to remind us, this takes time and it takes people. And I am really grateful that we're able to be here today with the FDA-approved study. We briefly talked this morning about the open label extension. So that was the extra 40 weeks that people could enroll in. That was what they called LILAC-1. When people completed that 40 weeks, they could continue on in what they called LILAC-2. So both the LILAC-2 and then Daffodil, which was the study for 2 to 5-year-olds, those are ending now that it's commercially available. They're getting everyone in for their last study visit. So that when those studies end in June, then we'll learn more data on this long-term use, so beyond the initial 52 weeks. Some of you may know about Anavex. So Anavex is another drug. Anavex 273 is a drug made by a company named Anavex, and it's a sigma-1 protein agonist, which makes sense to everyone, right? What they say is that it promotes neuroplasticity and restores neural cell homeostasis. Jargon, right? You know, what does it really mean for my kid? Will my kid walk, talk, use their hands? We don't know. But we do know that the brain has neuroplasticity, meaning it can change, right? So we go from being infants where we can't talk, walk, dress ourselves, to with repeated exposure, our brain, changes so that we can learn these tasks. And with Rett, we think it's still the same. There's not an age cap where you're like, oh, well, after 1, you can no longer change, right? I mean, don't we still change as adults? So the brain is plastic, and so drugs that impact neuroplasticity, are expected to have some benefit in Rett. So they're doing the clinical trials. They did their first clinical trial in the U.S. in adults in 2019. And then they also started an international adult trial around the same time, a little bit later, but in 2019. Both of those studies have completed. They then completed a pediatric study that was international, so not in the U.S. So that just completed earlier this year, and they said they're going to share the data in the second half of 2023. So we don't have updates yet, but from the first 2 studies, which were both 7-week studies, they did demonstrate that the RSBQ that we heard about earlier today and that CGI-I that we heard about earlier today, both of those showed improvement. So we'll look forward to hearing an update from the company later this year. So those are the 2 drug trials that are, the furthest along. There are other drug trials that are going to be coming in 2024, so companies that are getting ready to launch their clinical trials. We have an exciting announcement coming up yet. You guys can be the first to hear it. I'm not going to tell you all the details, but we got a big DoD grant for a new clinical trial testing multiple different drugs in Rett syndrome that the trial itself will start in late 2024, most likely. So we have more drug trials that are coming down the pike in 2024. I don't have enough details to give you guys kind of the details of them, but I'm excited about those that are coming forward. So then switching to genetic approaches. This has been something that ever since for those of you who haven't heard it, people call it the reversal study, where Dr. Adrian Bird's lab demonstrated that in the mouse model of Rett, if you can give back the gene at some point in development, that the symptoms of Rett reverse. That really was a pretty big paper because it showed that this isn't degenerative. You could regain skills. It's not like some of the other neurodegenerative conditions where once you've lost those skills, those neurons have died and you cannot get them back. So that's why we say it's a neurodevelopmental disorder. It's not degenerative. And so that really opened up the window to, well, let's fix that gene. Let's turn that light switch back on. Let's get that gene back. And it's not as easy as that. Back in 2007, there was a lot of enthusiasm about it. And I'm glad that all that research has been done because we are where we are today because of more than a decade of research in how do you fix that gene in Rett. So just a broad overview. I'm only going to touch on 3 different genetic approaches for Rett because these are kind of the ones that are the most advanced. But I just want people to know the language. So gene therapy is sometimes used as an umbrella term for kind of all genetic approaches. And sometimes it's specifically used for a gene transfer therapy. And what this is you are actually giving a gene back. So if you have a faulty gene, faulty MECP2, you're injecting a working MECP2. So gene transfer, you're giving that new gene back. This is a one-time treatment. So I think Dr. Feynman touched on it a little earlier today, where you have this, I think of it as you've got your truck, and it's got its load in it, right? So your truck is that virus. For conditions of the brain, they use a virus called AAV9 as of today. That's the best virus we have that knows how to get to the brain cells and can actually get there. So your truck is your AAV9. And it has a load in it. It's carrying stuff. And what it's carrying is the MECP2 gene that you're going to put into the cells, right? So you've got your truck, and it's got MECP2. But we know with MECP2, too much MECP2 causes MECP2 duplication syndrome, too little causes Rett syndrome. So you need to have just the right amount. And so we'll talk about 2 companies that have gene transfer therapy protocols that are on, that they each are testing out a way to regulate that. So that's also in the truckload. So you've got your gene, you've got this regulatory piece that's going to try to help control the level of MECP2. And then it has to bring other stuff with it too. It's got to be able to give some of the machinery for this to work. So that's all in your truckload. So what it does is you give this one-time treatment. The virus goes where, hopefully it goes where you want it to go. For Rett, we want it to go to the brain. But it also, as Dr. Feynman mentioned, it likes to go to the liver. And so there are side effects from these. Because you're giving a virus, your immune cells see this as a foreign object, because it is foreign, and their little soldiers then create a memory to it. Just like when we get the flu, or when we get, some other virus, your immune system builds these memory cells so that the next time your body sees that virus, it attacks it. Which is why with the current technology today, you cannot give gene therapy more than once, because your body would attack it and kill it before it could work, because it's that viral delivery. So it's a one-time treatment. So another type is something called RNA editing. So I describe RNA as your short-lived cousin of DNA. So DNA are your genes, they're your instruction book. They tell your body what to produce, something called proteins. And your RNA is kind of the middleman. So he has to take your DNA, it becomes his RNA, and then it goes to your protein. That's like a super simplified way. But the RNA is short-lived, so it's not permanent like your DNA. But you can change your RNA and still get that MECP2 protein correction without changing your DNA. So your DNA still has the error in it, but your RNA, the middleman, gets corrected, so then your protein is made. But it's short-lived, so you have to give it multiple times. And you can, if you're not using that viral delivery, these RNA editing techniques, you can give them more than once. The other thing about the RNA editing is that because you're not giving the whole gene, which when you give the whole gene, it doesn't matter what mutation you have, because you're giving the whole thing back. For RNA editing, they build it specific for a particular mutation or a mutation type. And so the RNA editing techniques will not be relevant to all types of mutations with Rett syndrome. Then there's another approach called reactivation of the silent X chromosome of your silent MECP2. So in women and girls with Rett, we have 2 X chromosomes, and in every cell of your body randomly puts one of them to sleep. It gets silenced. So in every cell of your body, you only have 1 active X chromosome. But there is that other silent one still in your cells. And so there are studies that are being done to figure out how to turn that silent one back on. And if that silent one carries the mutated MECP2, it doesn't matter because it's not working. And if the silent one carries the good MECP2, well, then you got more MECP2 back. So that's an approach that's not yet ready for clinical trials. Neither RNA editing nor reactivation are yet ready for clinical trials, but those are coming a lot closer to clinical trials. So I would say in the next 5 years, we'll see those. So the two trials for gene transfer therapy or gene therapy, the first one is from a company, Pichard, and this is what they call a phase 1-2 study. So when we do drug trials, so like the trofinetide trial, we started directly with the phase 2 because it'd been tested in humans before. Typically with drugs, you do a phase 1 healthy volunteer study. So we say, okay, you 3 healthy volunteers, take trofinetide, we're gonna study the side effects from it, right? But with gene therapy, you can't say, okay, you 3, you signing up for gene therapy, we're gonna inject this virus into your spinal fluid and see how you do. You can't do that, right? So your first in human study is in the population that you intend to treat, or for us, your Rett syndrome patients. So they call it, this is a phase 1-2 because it's the first time this product is being used in humans, and they're also looking at the safety and what the effect of it is, what the benefits are, and that's the phase 2 part of it. So Pichard is recruiting adult women with Rett syndrome in Canada right now. They do plan they're working with the UK to open the trial there. And next they're talking to the FDA to open the trial here in the US. Right now it's for individuals that are 18 and above. They said that they're looking to modify it to make it 15 years of age and above. But right now their study, it's 12 participants in Canada that are 18 and above. This is open label, which means there is no placebo group. Every single one of them will actually get the real gene therapy. But Pichard is looking at 2 different doses, which means part of the group will get, I'll call it dose A, and part of the group will get dose B. If you were in dose A and it didn't work, you can't get dose B because you can only get this one time. But they need to study what is the right dose because these are all experiments. We can model off of animals, but we won't know until it's in human. I mentioned it's one-time delivery for this one. They give it like a spinal tap. So it goes into the fluid that bathes your spine and goes up and bathes your brain. And their study is a 52-week study. So you're followed, usually gene therapy studies, you're actually followed for 5 years, but the 52 weeks is where they're gonna be able to say, okay, this was the benefit. Like trofinetide was 12-week study, but they continued to follow people. This will be a 52-week study. The next one is Neurogene. So this company also has a similar, gene therapy product. They also have that regulatory element, but this study is going to start in the U.S. in children. So this will be girls who are aged 4 to 10 years. Also a phase 1-2. So even though Pichard is doing a first in human, this is a totally different product, right? So that whole truckload that's in the truck is different, which means it's the first time this product is in a human, different from the Pichard. So they have to do a first in human for this too. So they have not, they haven't started enrolling yet, so they haven't made as much information publicly available yet. So they haven't announced what the clinical trial sites will be. And this one is also open label, which means everyone that goes in the study will get active gene therapy. And this one-time delivery is actually directly into the brain. And so it's a common mechanism for neurologic disorders to do direct injections to brain. So it's a neurosurgery procedure. The doctors that do it are very skilled in this. They view it as like, oh yeah, like I get up and brush my teeth every day. They do direct injections into the brain every day. They think it's pretty, routine. So just to, rest assured that this is not done by someone that's never done this before. These are experts in delivering directly into the brain, but it is a neurosurgical procedure. So one of the things that came up recently, and Dr. Feynman mentioned it earlier today, Neurogene did put out some basic information about their trial and did mention that any individuals who've had any exposure to trofinetide would not be allowed in their trial. We promptly got on the phone with them. We mobilized our Center of Excellence network and asked them, what's the scientific rationale for this? These are experiments. These are first in human. There's a lot, to understand. But, why would being on a drug 1 to 10 years ago influence your ability to be on a gene therapy, which is a totally different mechanism? And what they've revised their communication to really clarify. What they meant was the first few patients cannot have had exposure to trofinetide because this is a first in human. They want to be 100% certain that anything that's going on is due to their product and not some leftover residual effect from some other drug. And they also stated specifically that after the first few, trofinetide exposure will be allowed. So it is not an exclusion. And so I would not worry about any future clinical trials. If you're like, oh, but I want my adult child to have gene therapy when it's available, should I not take trofinetide? And the message is, this will not be an exclusion. Of course, this is a clinical trial. It's first in human. But there's no direct interaction that would make it concerning. All right. I do want to say, so this top resource that I put up here, the American Society of Gene and Cell Therapy, they have a part of their website for patient education. They do a very good job of explaining what gene therapy is, explaining that direct-to-brain injection, the spinal type. So I think it's a good resource to learn about gene therapy if you're interested. It is the society talks about all sorts of gene and cell therapy, so there's some stuff that's not relevant to us. Because a lot of the blood disorders, their gene therapy looks different. You actually can take the blood out, do the gene therapy, and then give the blood back. We don't take our brains out and then give them back. So we don't do ex vivo gene therapy, we do in vivo, like it happens in the body. So there's some of the stuff that you might read and you'd be like, wait, she didn't talk about that. Well, it's because it doesn't really apply to Rett syndrome. And then I just put on here 3 different IRSF websites or links. So we have a Rett Research Ready page, and this was really designed to help individuals learn about what is clinical trials, kind of what are the questions you might want to ask if you're considering a clinical trial, what are some of the terms that are thrown around. And so I invite you to look at the Rett Research Ready website. We also have on there a trial finder, so you can look for research opportunities. These are clinical trials, but also like the studies that are done here in Minnesota. As we try to load those in there, so you could say, hey, I'm willing to do what works for my family now is to do a survey-based study. I'm going to see if there's one that's relevant that I don't have to travel for, that my child would qualify for, so I can contribute to the research. Or you say, okay, I'm ready to sign up to do some eye gaze studies. Let me see what's going on right now. And so we try to keep that as up to date as we can, as we know about research that's going on that's not clinical trial, but it also has all the clinical trials that are going on in the US. And then finally, we update the clinical trial news section. So as companies release any statements to the community or press releases, we put them on there so that you have one place to go to learn about all of the different clinical trials. And that is really it. I want to thank you for the opportunity to be here today to talk to you. I am happy to answer any questions. I'm here all day. So whatever time we have for Q&A is great. And otherwise, you can find me later. Thank you.
Well, it's fresh on your brain. Let's fill 10 minutes of questions, but she's sticking around, so lunch. Good luck eating a lunch, Dominique. You want me- all right, questions. If you have one, I just need you to shout it loudly because our mic is picking it up, but I just need it to be clear. So, any questions? Oh, come on, John.
I'm a lay person as far as gene therapy. That seems to be just intuitively a little bit more promising than maybe the drug therapies as far as, you know, a cure type of thing.
Yeah.
I should look at that one-on-one thing that you have, but how long has gene therapy been around, and is there, you know, other diseases or symptoms that it's been able to cure? I guess I'm just not aware of that.
That's a great question. So gene therapy first came into clinical trials in the 1990s. Dr. Feynman mentioned kind of this. In 1997, there was an individual who had an immunodeficiency who got a gene therapy at University of Pennsylvania and died, which kind of put a big damper on the gene therapy field. And so the research slowed down quite a bit in the human translation of gene therapies because everyone was so scared. That individual died because of the immune reaction to the gene therapy rather than, you know, the disease itself or something about the product. So it was just the body kind of went crazy. Because of that, they've learned a lot. Gene therapy has come a very long way. It is much safer in that sense. And you're right, it is the most promising approach to have the greatest impact, right? Because when you get the gene back, you have the highest prospect for most benefit. But what we don't know, you know, while the mouse, which is about this big, was able to reverse its symptoms, even when it was a very late stage Rett mouse, they did this trick where basically in mice, you can do, you know, genetic editing. So they turned the MECP2 gene off, and then when the mouse was like really sick, really had like full-blown Rett, they could turn the gene back on, and bam, you know, they reversed most of the symptoms. It was not, it looked pretty close to normal. It wasn't 100% normal, but that's pretty amazing. That doesn't really recapitulate what happens in our kids because they didn't have like a gene turned off before they were born and then turned back on at some point. You know, some of our kids have, you know, we do have one functioning X chromosome with some MECP2. And so it's a little bit different, but there's hope because of that. So it is the highest prospect. It's also early in its studies. You know, we're doing these first in humans. When you get gene therapy, you have to be on immunosuppression. So because they need to, so that we don't get the reaction that happened to Jesse. So you're on immunosuppression, you're getting this product. Is it the right dose? Did it actually get to where it needed to be? Did enough of it get to where it needed to be? Did it last long? So those are all the experiments. That's why we do clinical trials. So in other conditions, the closest to Rett is something called spinal muscular atrophy. And so this is a neurodegenerative condition. And in their most severe form, the type 1 SMA kids, they only live up to usually around 2 years because their neurons degenerate. So they lose them. And once they're gone, you cannot get them back. And they did a clinical trial with gene therapy, and those kids lived. So now what they found is, again, it's a very different situation because it's degenerative. It's FDA approved for kids 0 to 2 years of age that have this type 1 SMA. The younger they get it, the better they do. Are the kids who got it totally typically developing? No. But they're living, they're sitting up, they're eating, which are not things that they would have been able to do with SMA without the gene therapy. So that is one of the, you know, that's like the most promising and, you know, what got everyone excited is when SMA had this approval, it's like, this is possible for a neurologic condition. But every condition is a little bit different. So there's a lot of gene therapy studies going on. The part of the FDA that reviews gene therapy, the number of applications for clinical trials has skyrocketed. Like they're having to increase their staff exponentially to review all of these studies because there are so many gene therapy studies going on. Dr. Feima mentioned muscular dystrophy. There's a lot of gene therapy studies going on for muscular dystrophies. So it is going on in many conditions. The ones that have been approved have been the SMA, eye disorders, which is a little bit different because the eye is a little bit more accessible, some of the blood disorders, and so for neurologic, it's only SMA.
I'm going to pull rank because I have a question. I've never thought of this, but where do they get, okay, they're going to truly put this in a human being.
Yes.
Probably in Canada as we speak, they're screening someone. Where do they get the gene? I'm assuming it's not synthetic. Where are they getting it? And I'm assuming it gets screened that it's really a healthy one? So,
Yeah, so it is synthetic. So they've got to make these, right? Because they can't say like, oh, hi, can I have your MECP2 gene? I'm going to test it. Which really just means that they have to make it. So it's something that is made in the lab, and the whole truckload is combined. So the Taysha product, they're actually testing what they call a mini MECP2. And so one of the labs studied and identified that you could make a MECP2 gene that's smaller, which means, if you think of like your truck, you're not getting to your weight limit with the gene alone, which means you have room for that other stuff that's important. So they're using a mini MECP2 gene, which is synthesized, which allowed them to have room for the tool that they're using to control the level of MECP2, which was bigger. Neurogene is using the full-size MECP2 synthetic product, so it's something that they have to make.
all right, how about one more question? Come on, stump the scientist. That's hard to do, yes.
I was just curious, what for the study in Canada, what requirements did the girls need to be able to be in that study? Were they really severe cases, or just?
So, top of my head, I don't remember all of their criteria. I know that they have to have a mutation in MECP2 because you're giving a MECP2 back. They had to meet criteria for clinical classic Rett syndrome, so it did not include atypical Rett syndrome. As I mentioned, 18 and above. I don't remember what severity scale what severity score they used. Yeah. My recollection is that they're not the most severe. They had to be severe enough, but it is not. In some studies you see you kind of do the most affected or the sickest or first, but they were able to get this with kind of more aligned with what we saw with drug trials. So if you, our Rett Research Ready website does have a link to it, but also another source, which I didn't put up there because I don't find it to be as family friendly, is clinicaltrials.gov. So it's www.clinicaltrials.gov, and then you put in the search bar, the condition is Rett syndrome, and you can put in Taysha if it'll help narrow it down, but you could find the clinical trial there, and what it shows, it will actually list out all of the inclusion and exclusion criteria. So if you were interested in understanding, because sometimes there's like certain lab values that will preclude you from being in, like if you have any liver issues, you're not going to be in a gene therapy trial. If you have, so there's a lot of criteria that go into it. So they're all listed on that site.
All right, well, we are going to wrap it up, but again, day is not over. Dominique.